हेलो, गुड इवनिंग लक्ष्य। एक्सप्लेन लिंकेज एंड रिकॉम्बिनेशन। तो तुम्हारे इससे कोई स्पेसिफिक क्वेश्चन हो तो बताओ। यस यस यस, आई कैन टीच इन इंग्लिश। आई वाज आस्किंग इफ यू हैव एनी स्पेसिफिक डाउट फ्रॉम दिस टॉपिक और डू यू वांट मी टू एक्सप्लेन दिस फ्रॉम द स्टार्ट? Okay. So see linkage and recombination. So this is a physical phenomena. Okay, that occurs inside the chromosome of our DNA. Okay, physical phenomena which is present on the chromosomes. Before moving further, just tell me, do you know about what is meiosis and mitosis? So you must know about the crossing over process that takes place in the Pachydian stage of the prophase 1 of meiosis 1. Do you know that? Okay, good. See, linkage. What do you understand by looking at this term? Linkage means some things that are linked. Okay, that are linked, and if you move one, then the other will also move with that because they are linked. Okay, isn't it? This is the literal meaning of the linkage that occurs inside the chromosome. So, linkage, it is the tendency of the linked genes to stay together and go inside a gamete. So generally what happens? In a diploid organism there are two sets of chromosome, one from the father and one from the mother. Parental are both. Okay. So this is not correct to say parental. Yes, you can say. No problem. So just for an example, let's understand this. This is a chromosome. Say it is chromosome number 11 and this is another chromosome number 11. One from is the father side and one is from the mother side. Okay, I am not making the two chromatids here, only one chromatid is sufficient for your understanding. Okay, we are not going on how the exact mechanism is occurring, we are just understanding. Okay, just to understand. Okay, so this is, let's take a certain gene such as HbA1. Okay, then it is a homologous chromosome. Okay, if there is a HbA1 on the chromosome which is coming from the father, then there will be absolutely one more gene which is present in the mother or the maternal chromosome. Okay, these two will be called as the alleles, isn't it? You know about alleles? Yes. Now consider there is one more gene here, HbA2. Now you can see that the distance between these two genes is very less. Okay, or you can also say that these genes are linked. Less the distance, more the linkage, isn't it? So I am writing here, linkage is directly proportional, sorry, inversely proportional to the distance between genes. More the distance, less the linkage. Less the distance, more the linkage. Okay, distance between genes. Okay, now Consider one more gene which is present here. This is HbA3. Okay. So you can say that the distance between HbA1 and HbA3 is very high. Okay. So what happens now during the gamete formation, meiosis will take place, and in case of the meiosis 1, prophase 1, okay, in the Pachytene stage, what will happen? There will be crossing over. What is crossing over? You know that? Yes. In crossing over, very good. Exchange of genetic material between what? Yes, between the homologous regions of the homologous chromosomes. Okay, that will be the correct definition of the crossing over. So, what will happen? Crossing overs will take place, and just remember, if you don't know this, crossing overs will take place at hundred of places in the homologous chromosome, not one. Many students think that crossing over will only take place at one end and the other end due to the diagrams given in the NCRT of the class 11. It is not like that. Crossing over can take place at multiple sites. Okay, just think like that. These are the crossing over sites. Crossing over can really occur at a lot of sites, more than 1000 or 2000 sites. It just depends. Nobody knows even today how it is that it is controlled. So we do not need to go in that. It is something which is control of the god. Okay. So just consider, if some genes have less distance, the possibility of the crossing over here will be very less, isn't it? And the tendency that these two genes will be separated is very less. And if you consider the distance, if you consider the genes HbA1 and HbA3, there will be a lot of crossing over between these two. You can say that between HbA1, I am writing H1, and H3, these two, approx 1 2 3 4 5 6, approx 6 crossing overs are taking place, and between H1 and H2 only one crossing over. Okay, so this is what is linkage. So if there is more linkage, there will be less recombination between the two genes. Recombination is directly proportional to the crossing over. If cross Recombination means the formation of the new genotypes. Okay, if something is linked and it is moving forward in the same way, there is nothing new coming, isn't it? That will be same in case of the parents, and also same in the case of the daughter cells or progeny. Okay, isn't it? But if something is being crossing over, okay, if crossing over is taking place at certain genes, then that will be a new type of genotype, or a new type of genetic combination will be there. So that is called as recombination. So crossing over sites are also called as recombination sites. So I think you know that they are synonymous. Okay, recombination sites. Okay. So what was I was yes, yes, ask, what are you asking? See, the purpose of linkage and recombination, there is no purpose of linkage, okay? But the recombination is a very good thing because it is giving new type of genotypes, and that new type of genotype are increasing the genetic pool. Do you know what is genetic pool? Consider something, some type of pool, swimming pool or any pool, okay? And there are multiple type of genotypes. Some genotypes are of blue type. Consider this, just it is an example. Some are of green type. Okay. Some are of white type. I think that you are able to differentiate between the colors. Some are white, some are yellow type, and some are pink type. Now consider there is a catastrophic event that occurred here. Catastrophe, tsunami, earthquake, forest fire, any kind of catastrophe, floods, etc. And that catastrophe removes selectively the pink type of individuals. You can see I am removing the pink type of individuals. Okay. Still, still there are individuals which are white, blue, yellow. So they are surviving. So gene pool is nothing but the collection of variability. Collection of variability. This is called as gene pool. Have you understood? So, so recombination increases the variability within the population of a species. Okay. So this gives the benefit in evolution. Okay. So just consider there is nothing harsh condition happening right now. But what if unnaturally or naturally or due to man-made reasons something happened and there is a lot of deaths of human population, then consider this death of population due to high temperature. You are seeing the temperature is rising very much. Consider after 10 years the temperature rises to 60 or 65 degree. I am giving you I am saying in 100% confidence that approx 90% of human population will be gone if the temperature is that. Okay, 65 degree or 70 degree. But still, 90% will be gone. The 10% population will be heat resistant. There will be some variants currently present right now which are heat resistant. They will survive. And how they appeared? Due to this chance variation, due to this random recombination process, there is no use of them right now, but they may survive during any kind of condition. Something now think there is a type of cold, more cold happened here. Okay, -10, -15. A lot of individual will die, but some will remain unaffected. Okay, so those population will be cold resistant. Every type of variety exists in the humans. It depends on the nature when it gives them the chance to grow and reproduce more. That is called as natural selection. Okay, natural selection can happen any time, and these gives the benefits, and these benefits arises due to crossing over. Have you understood or not? This is the beauty of the sexual reproduction. That is why sexual reproduction is more beneficial as compared to the asexual reproduction, because sexual reproduction is giving chances for the adaptation. Yes, variation that will lead to adaptation, and adaptation will lead to the natural selection and to the survival of that organism. Now come back to the topic about the linkage and recombination. Linkage is not giving any kind of benefit. Okay, it is just a phenomenon. Okay, and linkage is just exactly opposite to the recombination. So linkage is inversely proportional to distance between genes. It is inversely proportional to the recombination. If there will be more recombination, there will be less linkage. Okay. So now these in the NCRT book, if you have the NCRT book, they have a very good like what we say that there is a beautiful cross of the Drosophila. There is a XX chromosome and then XY chromosome. Can you send me that image if you have the NCRT? That will be like this. These 3 to 4 blocks, this type of graph. Are you understanding what I'm saying? If you will send me that image, I will quickly tell you what is that. Yes, very good. So now see in this image, these are two separate crosses. So I will come into the cross B later on. Now focus on the cross A. Okay. See, in case of the cross A, the parents. Okay, parental means the parents. This is a female having XX chromosome. Okay, and this is XY chromosome. Okay. Now see there are genotypes YW and YW. There is not plus sign on them. That means it is a recessive character. Okay, and Y plus W plus in case of the male. So you know how the gametes are formed. See this female will Y plus W plus means they have the dominant trait of that Y gene. The plus sign means dominant, and with no sign it means recessive. Okay, that is why it is yellow white. So you can say in case of the what is that phenotype? I think it is eye color, maybe. I'm not sure. So just remember it is the dominant and the female one are having recessive. So both of the X chromosome is having YW. So the gamete will have YW in case of the female, isn't it? Only one type of possibility is there. But the male will produce both type of X and Y, both type of gametes. Okay. So one X chromosome will go with the X chromosome of the male and it will result in this phenotype, and one X chromosome will move to with the Y chromosome of the male and this will produce this phenotype. Have you understood? Any doubt in how is this cross is being formed till here? You didn't just understand how the F1 formed. See, I am just forming a Mendelian cross. Don't look into the image. The female is having YW and the male is having Y plus W plus. Okay, this is one possibility. This is one chromosome and this is one chromosome. Okay, it is one sex chromosome. Okay, so consider this as, no, let me write it as, this is female, this is male. Female having X and X, and male is having X and Y chromosome. Okay, so the X chromosome of the female, the X chromosome of the female is having YW. Okay, and this X is having YW. Okay, and the male X chromosome is having Y plus W plus. Okay, and the Y chromosome is having no gene, because you know that the Y chromosome is not homologous with the X chromosome. So there is no gene which is given here, so we will not consider, we will not also consider Y chromosome. So YW can go with this, isn't it? And this can go with this. You must know how the Mendel formed those. Okay. So now the possibility will be YW going with Y plus W plus, and this will be YW going with Y chromosome. I am writing it as like this, and then again it will be similar, YW going with Y plus W plus, and it is YW going with the Y chromosome. So you can see that these two and these two are exactly similar. So your book has only mentioned one. Okay. I hope now you have understood this. Now these two are being formed as female and male. Now these two will be crossed. Okay. Now what will happen? You can again make the cross in that similar way. See what can happen. This YW will go with this YW. Okay. And this YW can go with this Y chromosome. Okay. And similarly this Y plus W plus can go with YW, and this Y plus W plus can go with this Y chromosome. Okay. So in total 4 type of possibilities in here and here. This possibility is for only the male and this is for the female in case of the F2 generation. You can see what all are the possibility with the male, that the Y chromosome can go with, also I have already formed, I don't want to confuse you, this Y can go with Y plus W plus, here it happened, and this Y and this Y can go with this YW. Okay, only these two possibilities, where it is, these two possibilities are the parental type. But see these two possibilities, these one, let me do it with yellow color, these two, you can see they have the new type of genotype which is not present in the parents. They are having Y plus W. Y plus was only with the W plus, but here Y plus is along with W and Y is along with W plus. Are you seeing some changes? Even the color change is also indicated that here some part is blue and some part is yellow. Can you see? And now see what is written in the above, the recombinant types. So what happened between the genes between Y plus W plus, the recombinant happened some at some places, and that recombinant frequency is 1.3%. You can see recombinant frequency is very low. It means if you will produce approx 1000 individuals, if 1000 individuals are being formed, only 130 individual will have, sorry, not 130, only 13 individual will have the recombinant phenotype, and all 997 individuals, or what will be 987, no? The 987 individual will have the phenotype with the parent. So this parental type means the phenotype which is resembling with the parents, and the recombinant type means the phenotype which is resembling with the none of the parents. Okay, recombinant resembles with none of the parents, they are new. Okay. Okay, so this is here, and it is similarly you can also see here, these are the recombinant. And here in case of the cross B, the same thing is being mentioned. I think the session will end soon, so if you want you can also join after this. There is some little thing, 1 or 2 minutes I will take extra. So have you understood this crossing? Now see, in cross B everything is same. These are the parental, this is F1, and these are the F2. This one is male and this is for female. Now see here, the recombinant frequency is 37.2%. Okay, and parental type is 62.8%. So that is the only difference between these crosses, only the frequency is differing, the rest all are the same. Okay, and one thing you can see that initially these two genes are very closely located, but here you can see these two genes are very much far apart on the chromosome, isn't it? So they are far apart, the recombination frequency is more, isn't it? Because number of